Good morning, everyone. I'm going to talk on a topic which is spray-induced gene silencing. Spray-induced gene silencing as an environmental RNA interference for the controlling pine pitch canker. Well, as a forester, we all know about the pines, and you know that pines are very important for many purposes, for the timber production and also for the economic purposes. So for this, we need to focus on how to control different diseases which is happening in the pines. So one of the most important pathogen which is on the pine is Fusarium sarcinatum. Okay. It is not only affecting the pines, but it is affecting more than 100 plant species, including some herbs and some grasses. Talking about the distribution of it, it was originated from the Mexico, and then it spread in some regions of USA. But it wasn't present in Europe, but later it introduced in some countries of the Europe, including France, Portugal, and Spain. And you can see here, it's still present in Spain and Portugal, and we can't control this. So how it spread? There are different means of spreading. First of all, the seeds and the wind. These are two main sources of inoculum of the pathogen. But it can also spread to insects as a vector and human transportation activities. Well, talking about the symptoms, as we all know that trees develop from the seeds. So nurseries are the main factors of this disease spread. In the nurseries, we have seen that we have different damping off of the seedlings. We have the complete browning, and almost the seedlings die back from upward to the downward. And basically, in the nurseries, it is asystematic, means it's in a lot of infection. So if we can't control in the nurseries, it's going to be infected in the trees, and we lost everything, almost. In the mature trees, it's caused what? Like yellowing of the needles at the upward, the canker formation, and the resident exudation. So we are trying to control it with many ways, like biological control with different phytosanitary management, but up till now, we can't control it at 100% rate. So for this purpose, we have to find a good, promising tool, which is also environmental friendly, because we can't use the fungicides due to different legalization problems, especially in Europe. Okay. Now I will talk about spray-induced gene silencing, but first of all, we all know about what is central dogma, because we all, most of us from the biological background. So we have the DNA, and then this DNA is going to form mRNA, which have all the information of the genetic, and then it's going to form the protein, which have the regulative functions in our body. So what happened in the spray-induced gene silencing? We are going to apply an external dsRNAs, which we are going to form by ourselves, and then we are going to spray it in the pathogen. What happened? They are going in the pathogen, and in the pathogen, they have some machinery, which is we call as RNA machinery. In the pathogen, in mostly now the eukaryotes, they have present, and it is conserved. So what did this machinery do? It's cleave our dsRNA into the small interfering RNAs. And these small interfering RNAs, what are they going to do? They try to target the messenger RNA, the pathogen. So it's bound with that and block the messenger RNA and block and degraded it and split it in different pieces. It means we don't have proteins, and we don't have protein, it means we are not nothing is going to express. So this is basic mechanism of spray-induced gene silencing. Well, this is the first work that we did in our lab with the help of my two supervisors. One is Julio and other one is Jonathan. So I'm glad, and we are going to present, because we didn't work on, no one is working on this, so we have to find out the genes which are very essential for the Fusarium sarcinatum. For this, we have to study the literatures related to different fusariums. And what we did, we studied different pathways in fusarium and other botrytis which are affecting different other pathogens. Because fusarium sericinatum is the species which is originated from, it's complex, and we can find it how we can control that. For this purpose, we selected 3 different pathways. The first one is vesicle taraphine. And for this pathway, we selected 3 genes. Why we selected 3 genes? For most of the pathways, we selected more than 3 genes, so we can silence more genes at a time, and so we have more effects. For the cell wall, we selected almost 4 genes, and for the signaling, we selected almost 4 genes. And for the negative control, we selected just 1 gene, and it is basically untarget specific sequence. It wasn't related to the any gene, it's untarget, so we can compare these two. Well, before starting that, I have to know, either my fusarium sericinatum, which I'm going to plot the dsRNAs, is going to uptake or not. So for this purpose, we synthesize our dsRNA, which is fluorescence. We give in the signaling glands, and then for the spores of the fusarium sericinatum, we try to germinate in the 2 different sporulating mediums. And then we inoculate both together and wait for 3 hours so that the fusarium can uptake the dsRNAs, and at the last, we measure the confocal microscopy. Well, what we see? As we see, we have one control, that is YFP without any dsRNAs, we didn't given signals. And in this one, we have the labeled dsRNA, and we can see the green lines. It means our pathogen is going to uptake the dsRNA. So this is our first success. Well, we have to produce these dsRNAs when we know, okay, this is going to work. So we construct this dsRNAs in the cloning vector. Well, for this we use 2 plasmids. First, we use T7 plasmid, which has the 2 promoters. 2 promoters means it can produce 2 dsRNAs, 2 RNAs, which is complementary to 2 dsRNA strands. So we have 2 strands of dsRNAs, one complementary to each other. And then we introduce it into E. coli, and E. coli is basically RNase 3 deficient. It means it's not going to chop up our dsRNAs. We have the intact dsRNAs. Well, after that, we have to produce in a bulk quantity because we need to apply in our different plant materials. So for that, we do it in the culture, LB culture, and then we produce it, and after that we extract the total RNA, and from the total RNA we extract the dsRNAs because we have a lot of type of RNAs, and so we have to purify these dsRNAs. Well, here you can see I didn't use pines because pine is my plant, but why I use something else here. So, pines take a lot of time to grow. So for this purpose of the food efficacy to the test or to test a lot of dsRNAs, we try to use different plant materials. We use tomato, spinach, and rose, and pines were good. So what's happened? In the left side, we applied our control, and then in the right side, we applied our treatments. And we evaluate at the 4 DPI, and we evaluate 2 things. One is lesion size measurement, and other thing was that relative fungal biomass. Well, our results was very good. We have a statistical differences between the results. First of all, in the left side, the control, they have bigger necrotic lesion as compared to the right side. And while in the relative lesion size, we have found in the reduction of different percentages, and in accordance with the relative lesion size with the fungal biomass, we also find the reduction of almost 54% and 60%. So it means our pathogen, which is fusarium sericinatum, can be controlled through dsRNAs. Well, next step was the 6 in the pines. For the pine assay, we take the pines which were in the seedlings, and why we choose seedling? Because nursery is the main causal agent to spread the pathogen. So we take seedlings, we cut this, and then we use a SIPSTER assessment using the Coral scale, 0 to 5, and then after that we did evaluation at 2 different time points, 25 and 35 DPIs. Well, here in the scale you can see the 4 and 5 is the most drastic one, because in the 5 point the tree is almost dead. Well, at the 25 GPI, I will show you some treatment, like we did all error test, and we all found that we have different type of symptoms, you can see. Well, water, it's fine. In mock, we didn't find anything. While our two controls, like what I told, YFP, and one is fusarium we use as a negative control, we have found like they have the mean disease score 2.5 and 2.38. While in the case of other treatments, we have found the disease reduction in 9 to 48%, while in the treatments, we have mean disease score 1.549 to 1.548. It means we have found something in reduction and growth. While at the 25 DRPA, we have two different measurements. Same in the case of water, we have nothing. It's 0. It means our milk doesn't have anything. While in the case of two positive controls, we have disease mean scale, which is higher. While all the treatments, they have 14 to 23% reduction. So what we decided that we are going to apply these treatments on repeatedly basis, 0 days, 7 days, and 35 days. So we increase the frequency of applications to know the effect. Maybe we have more effect on this. So in this case, we have the disease, in the case of this, we have disease incidence almost less virulent, and it's almost 25%. Okay, I'm just finishing it. And we have 3.55 MGS, which in the YFP case. Well, take-home message. First, we can use fusarium system is capable of uptaking exogenous dsRNA, so it means we can use it with other fusarium species, or if we want to control other pathogen. Other, I will say, like with the quick assay, we can use other plant material because sometimes our forest crop take a lot of time to grow, so we can use other alternative materials. And SIG is a promising tool, as we can say that we have seen it and it's giving us the almost same percentage as a fungicide is giving to control the pathogen. So I can say SIG is a promising tool. Well, I can say we use RNA-seq. I don't have a time to show that, but I also do RNA-seq, and I can say for the future we can use RNA-seq to find and to know how the plant is communicated with pathogen and to identify new microRNAs and new target for the SIGs, and we can use this also. Thank you so much for your attention.